Okay, looks good.
Good evening and welcome to our Haystack Rock, Discover Haystack Rock Library Lecture Series. I'm Angela Benton and I'm part of Friends of Haystack Rock, and we are so thrilled to have you here this year. The last time that we actually got to be in person and see real people in this lecture series was in January of 2020, so we're thrilled to be back in this space again. So thank you all for coming this evening, and for those of you that are on Facebook, we're glad you joined us. We are, tonight we're doing this lecture, and the lecture series this season is in partnership with the Cannon Beach Library, who graciously opens their doors and partners with us to have this incredible technology and technology support. So we're really happy to be here. Before we introduce our speaker for the this evening to kick off the 22-23 library lecture season, I want to spend a minute talking about this season. And this is a special season for us because this season is in honor of one of our Friends of Haystack Rock partners, fellows, volunteers, Sandy Lundy. This community lost Sandy, as all of you know, last summer, and she was really an amazing member of this community. And she leaves behind her husband, Bob, who has also been an amazing partner to us, many friends, and a daughter, Allegra. So, you know, Sandy was a special friend to us. She volunteered for Friends of Haystack Rock, very involved in the library. You could see her smiling face at Farmers Market, North Coast Land Conservancy. From my view of the community, wherever there was a gathering, usually Sandy and Bob were there. So they love people, and that's why, part of the reason why we're here tonight. And it was actually in 2008 that Sandy helped start the library lecture series. So this is really a way of honoring her. And both she and Bob, from my tenure and many other board members' tenures, they were always the first in the door. The place was always set up. They were always the last to leave. And there were always incredible goodies. So unfortunately, I've already failed at the goodie providing because they weren't homemade this evening. But really, Sandy and also her amazing partner, Bob, have been a force behind this library lecture series. And so we really felt like it was important to honor her and celebrate her life through dedicating this year's library lecture. Now, her smile, her laughter, her wit, her homemade cookies, and her tireless energy, I have to say, is going to be greatly missed, but we move forward in her honor. So welcome to the library lecture series in honor of Sandy Lundy. And thank you, Bob, for allowing us to honor her in this way. Just a little note about the library lecture. Many of you probably know about this, but the library lecture series is a monthly series, again, put on by Friends of Haystack Rock and the Cannon Beach Library. It is every month, the second Wednesday of the month at 7 p.m. And so put that on your calendars. We start in November, and our last library lecture is in April. So circle those dates, and our format, assuming all goes well this winter, will be to have both a live engagement, as we are here today, as well as Facebook, so folks can participate remotely. Now, one of the things we started doing last year was recording the library lectures. So if by chance your schedule is such that you can't make it, either physically or in Facebook, you can go to our we can go to our website and find the library lectures recorded there. It promises to be a great season. Thanks to Tiffany Booth and Jen Dixon for bringing all of this together, technology, schedules, etc. And with that, I'm going to turn it over to our board member, Young, and have her introduce our guests.
So good evening. I'm delighted to be here to welcome our first guest, Sylvia Yamada, who is a marine ecologist and a faculty member at the Department of Integrative Biology at Oregon State University. Her research includes salmon stock identification, predator-prey interactions, invertebrate fisheries, and the role of introduced fisheries and the role of introduced species in the marine environment. And she's been monitoring European green crabs in Oregon estuaries for over 20 years. And we're very excited to hear you speak about it.
Thank you very much. Yes. And I'd like to thank Tiffany for inviting me and Jen for helping me with the slide projection. First of all, I'd like to acknowledge some of my people that helped me in this study. Sean Schooler from South Slough National Estuarine Research Reserve and Jennifer Fisher of NOAA. Slide, please. And numerous other people in Oregon, Washington, and Woods Hole. Next slide, please. Okay, what do we have here? Here we have two crabs, a green crab and a Dungeness crab. And how can you tell them apart? They're both fan-shaped crabs. How do they differ? Any ideas?
Looks like the green crab is, and the Dungeness is flatter.
One of the easiest way to tell them apart is to count the spines behind the eyes. Now, the green crab has 5 distinct spines, whereas the Dungeness has 10. We can pass these around if you like. Another way is to look at the claws. The Dungeness has 2 more delicate claws than the green crab, has a crusher and a cutter claw. So it has a dual tool set. Next slide, please. Now, green crabs are known both as predators and as ecological engineers. Now, whenever green crabs are abundant, be it their native range or in their introduced range, when they're abundant, you can see a decrease in their prey, such as cockles, mussels, urchins, and worms. And perhaps the best example of what green crabs can do is the decline of the soft-shelled clam industry during the 1950s on the East Coast. The 1950s were a warm period, much like now, and the green crabs were thriving, and they were feeding on the soft-shelled clams. Now, the role of green crabs as ecological engineers has only recently been documented. What they can do when they're abundant, they can transform a productive eelgrass meadow like that to bare substrate. And the way they do it is they sift through the sediment in their foraging activities after clams and worms and other prey organisms. And in that process, they are tearing up the plants and they get carried away. And you all know that eelgrass are very similar to the rainforest trees and the corals on coral reefs. They're foundational species because many other organisms feed and find habitat around them. And for eelgrass, that would include a lot of fishes, waterfowl, shrimp, shellfish, and other invertebrates. Okay, the life history of the green crab. First of all, I have to tell you that the green crab is rather a medium-sized crab. It never gets more than 4 inches in carapace width. And here you can see a female with its rounded abdomen, a male abdomen like with a triangle. And you can see that green crabs are not always green. They're green right after they molt. They're nice olive green. But here you can see a little yellow on the underside, on the belly. And this one is orange. And just before molting, they tend to be red. Okay, so the color itself is not a good way to tell them apart. Count the spines, because there's no other native crab species on our coast that has 5 spines behind the eyes. And here you see a female crab carrying a batch of eggs. Now, the green crabs reach sexual maturity around 33 to 35 millimeter in carapace width on our coast. And they can only mate after the female has molted, when the female is soft. So what the males do is they're attracted to the scent of a molting female, and they'll try to carry her around and wait for her to molt. And when she's soft, then they can mate, and he'll carry her around for a few more days till she hardens up, and also to prevent her from mating with other males, of course. Next slide, please. Now, when the eggs hatch, you get cute little larvae. They're called zoea larvae, and they have really big eyes, and they have a huge spine to deter predators. And what these little zoea do is, when they hatch, they swim to the surface, and they ride an outgoing tide to the near shore, where they become part of the plankton. They undergo vertical migration, they feed on the plankton, they grow, and they molt through another 3 more zoea stages. And then the final larval stage is called a megalopa. And it's a transitional larval stage between the planktonic lifestyle and the more benthic crawl away lifestyle of a crab. What these fellows will do is they'll swim to the surface and ride an incoming tide back into the estuary, and where they will settle high on the shore amongst the vegetation. And then they'll molt again and become miniature crabs. Okay, so in summary, the life cycle of a green crab, like many other crabs, of course, has 2 life phases. It has the adult crab phase, and then it has a planktonic larval stage. Now, adult crabs, they can get carried around, in rock ballast and fishing gear and so on. But on their own, they can only maybe travel a few miles. But it's the larval stages that can be carried in ocean currents, over hundreds and hundreds of miles. So next slide, please. And how do they get around? Well, green crabs got to the East Coast, and also probably to Australia, in sailing ships as stowaways. They were, that sailing ships, they have to carry rock ballast just to give them a little stability. And many old harbors have places called ballast point, where ballast rocks are picked up and discarded. So a lot of other organisms are fouling organisms on the rocks, and that's how they got transported around. This is a exploratory oil drilling platform, and fouling on that platform is believed to be the mechanism by which green crabs were introduced into South Africa. Yes, and right now, green crabs got introduced into San Francisco Bay from the East Coast with Atlantic seafood products, probably Atlantic lobsters or bait worms from Maine. And what they do is they wrap it in seaweed to keep it moist. And then, of course, what do people do when they, come to their, if you're a fisherman, you have all these worms. You throw the algae in the water, and lots of other organisms have been introduced along with algae like that as packing material. And of course, once a green crab population is established, like it was in San Francisco Bay, then the larvae that it produces can get carried in ocean currents, and they can traverse hundreds of miles. Next slide. Okay, green crabs are a temperate species. You can see there are 2 critical temperatures that have to do with reproduction that somehow set the distribution of green crabs around the world. Now, and look in the native habitat, you can see they exist all the way from northern Norway, southern Iceland, all the way to Morocco and Mauritania. So they have a huge temperature tolerance, and also they're found in the very high saline Atlantic and also in the brackish Baltic Sea. Now the squares represent a sibling species to the green crabs. They're called the Mediterranean green crab, and they were introduced into Japan. Now, if the temperature never gets above 10 degrees Celsius or 50 degrees Fahrenheit, then green crabs can't grow, they can't molt, and the larvae can't develop. So that sets the poleward distribution. Also, female green crabs need cooler water, at least 18 degrees for part of the year, in order to successfully incubate their eggs. So you can see that all the green crab introductions are found in temperate areas. They've been introduced to Panama, to Madagascar, Sri Lanka, Burma, and Hawaii, but those introductions didn't take. It's only in the temperate region. This handsome fellow is a famous Norwegian fisheries biologist who over 100 years ago came up with the idea that of explaining fluctuations in the great fisheries of the North Sea. And these fisheries are, of course, herring and cod. And he made that observation that ocean conditions during the early life stages of these fishes can predict the catches a few years in the future. And for example, if the rearing conditions are warm, if the water is warm, then the young fishes, they survive well, they grow well, and you're going to have a good fisheries a few years later, and vice versa. And also, if there are some unfavorable currents that take the young fishes away from their feeding ground, you can have the loss of a whole cohort, a whole year class of fishes. So, and green crabs have been the same way. Temperature and currents are very important in their range expansion and also in their abundance, in local abundance in Oregon. Okay, okay, I mentioned there are two factors. Next, temperature, warm temperature, 10 degrees for larval development and 15 degrees for mating, for the adult green crabs to mature their gonads and to successfully mate. And next slide, and you need favorable currents. And next slide. That favorable current that explains the abundance of green crabs in Oregon and also contributed to their range expansion is called the Davidson current. It's a warm current that originates in Southern California, and it hugs the shore of California, Oregon, Washington, British Columbia, and then up into Southern Alaska. You can think of it as a conveyor belt or a river that's flowing, that's only in the wintertime. In the summertime, what happens is the wind shifts, the Davidson current dissipates, and the wind blows from the north, pushing water southward. That's in the summertime. And that causes the upwelling, and the upwelling brings nutrients to the surface and also brings copepods from the north that are really full of lipids, really good for salmon food. Okay, so summertime conditions with upwelling are good for salmon. Wintertime conditions with a warm current moving larvae northwards for range expansion into Oregon, Washington, and British Columbia. Okay, next slide, please. Here we look at San Francisco. You need to push, let's see. Oh, there we go, there we go. That's San Francisco Bay, okay? No. You don't need to push it. It will do it on its own. You just need to push the arrow. Yeah, just don't touch it. See, there's periods when nothing is happening. The populations are building up in San Francisco. Larvae are being, okay, right? There's a whole bunch of range expansions that happened during the El Nino of 97, 98. Not much is happening now. Now they got into the Salish Sea. Look at that. They're on Haida Gwaii, and now they're in Alaska. So you see that there are periods of rapid range expansions and periods of stasis. Next slide, please. And this slide summarizes it. These are you can't read it, but these are the years. So right here you have a whole bunch of range expansions from San Francisco Bay, Oregon, Washington, and southern British Columbia. And then right in here, you get into the Salish Sea, the inland sea between Vancouver Island and the mainland. And there's after 98 and after 2015, 16. And next slide, please. Those were El Nino years. Do you remember the 97, 98 El Nino when it was really warm? And then also the most recent El Nino, 2015 to 2016. Now, during El Nino events, see, these were unusually strong El Ninos. This was the strongest of the last century, and this one was just as strong. During El Nino years, the Davidson current during the winter is especially strong and of long duration. Water temperatures were up to 12 degrees Celsius or 54 degrees Fahrenheit. And the Davidson current lasted for September, October, November, December, January, February, March, April. 8 months you had the Davidson current being active. And, you know, like upwelling was delayed during that time. So there was a big window of time that larvae could have been transported from California to Oregon, Washington, British Columbia. And also the Davidson current was very strong, 30 miles per day for all of January, February, March of 98. Next slide, please. We do a little math here. In a non-El Nino year, the Davidson current off our coast in the wintertime is maybe 6 miles per day in the northward direction. And if you're a larva in the plankton and you spend maybe a couple months developing, you can travel, 360 miles. So in a non-El Nino year, larvae from San Francisco Bay could easily make it to Humboldt Bay. But in an El Nino year, you can travel 5 times as far, and you can travel all the way, to British Columbia, no problem. Okay, so that finishes the range expansion, the role of temperature and currents on range expansion. Temperature and currents were also very important in predicting the abundance of green crabs in Oregon estuaries and southern Washington estuaries. So after that 97, 98 El Nino, that summer and that fall, we saw lots and lots of little green crabs in all the estuaries that we looked at. And that year class was so strong, it was so well defined. There was nothing strong before it, nothing strong after it. Next slide, please. So what we did was we tagged a whole bunch of crabs. I got some students, volunteers from fisheries and wildlife and from zoology. And what we did was during winter break, we stayed at the Hatfield Center and we tagged a bunch of green crabs, and we used these little tags here. They're called spaghetti tags. They use them for, marking clothing. It's like a harpoon that you inject. And what we did was we would inject them into the gill chamber between the upper and lower carapace. So the upper and lower carapace has a line between it, like a suture line. And that line is the first one that dissolves when they're about to molt. So then it opens like a hinge, the upper and lower carapace, and the crab just crawls out, just like taking off a pair of gloves. And that is the beauty of this tag. The tag is retained. It's not caught up. And here is a red rock crab with such a tag right here. You can pass it around. Okay, so those are the sizes of the crabs when we released them. And then in July, we found some of them. And you can see that percent increase. The larger crabs, they added about 20 to 34% in carapace width. And that is characteristic of them having molted just once. But look at those little guys. They molted twice. So next slide, please. So using that information and also following the size distribution of the crabs as they made it through through time. It was very easy to do. We looked at Oregon mean, the Washington mean, British Columbia individuals, and then these lines here mark the extremes at the end of their growing season. You can see they grow very rapidly at first. The first winter, they reach 47 millimeters in carapace width. The second year, they molt it maybe twice, and then they molt maybe once, and then after a while, they don't molt, and they die of old age. Next slide. So 6 years is about the time that green crabs lived in Oregon, and that's the same as it is in Maine and also in Europe. So their lifespan is 6 years. So with no El Nino, and you know that green crabs only live for 6 years, then in 2004, we shouldn't have had any more green crabs, right? Problem solved. Next slide. Well, that's what I thought. Okay, 2004. Well, there's just a trickle maybe of a few larvae making it to Oregon. This is in Coos Bay. This is catch per unit effort. This number of crabs trapped per day, and these are the years. And in 2015, I went on vacation. I did not get to sample Coos Bay. I should have gone on vacation, right? But you know what? Look at the year before. I said 103 traps, and I didn't get a single green crab, or the year before. So there weren't any they were extremely rare. But then in 2016, 15, 16, we have another El Nino, and then each year they increase, and then this year looks like maybe they're leveling off. We don't know. It's too early to tell. Now let's look at this a little bit more closer. And here we are looking at recruitment. We're looking at the young crabs that are coming into the population, and we call them young of the year crabs. Now recruitment in marine ecology is defined as when the larvae settle on the substrate and metamorphose into whatever. But in fisheries, that's not practical. What they do is they call recruitment is when your fish enters your fishing gear. Simple, right? So in our fishing gear were these traps, and they enter these traps when they're about 30 millimeters in carapace width. When they're smaller, we get some, but usually not very many. So we waited until the end of their first growing season on the mud flat, like in September, October. We sampled these estuaries pretty much at the same time, as best as we could do it. And what you see is prior to 2015, it's hit or miss, right? There are a number of years, 1, 2, 3 years, 4, 5 years when there's no recruitment at all. We have total recruitment failure. And here you have some 2005, 2006, 2010. These were mini El Nino events, so we got some recruitment. And another thing to note is a good year in one estuary is a good year in another, and vice versa. So prior to this, we had very good evidence that the larvae were coming from California during the winter in the El Nino, in the Davidson Current. After the last El Nino, we get good recruitment every year. Next slide, please. Now, prior to the last El Nino, we looked at a number of ocean indicators, and they're related to temperature and northern transport. And what we found that any one of these would predict green crab abundance in Oregon and Southern Washington of the young year class. I will just give you two examples here, just plain temperature and southern copepod abundance. Now, southern copepod abundance means that there's a water mass coming from California carrying these southern copepods, and they're off our Oregon coast, and we measure them. That means that we've got a lot of water from California, and in that water could also be green crab larvae, right? So it's an indication of how strong the Davidson current is. The more southern copepods you get, the stronger the Davidson current was, and the more greater the chances of green crabs arriving in Oregon estuaries. Okay, here's a Davidson current again. All right, next slide, please. Now we superimpose water temperature, winter water temperature, on our recruitment graph, and you see prior to the last El Nino was a pretty good relationship. You know, when it's warm. This was the 97, 98 El Nino year class. It was warm. Right here, some warm period here. This was a mini El Nino. And after that, we get good recruitment every year, even when it's cold. Now, why? Next slide, please. Showing you the same information again, only this time we're looking at a regression of green crab, young green crab abundance and surface water temperature during the winter. And what we're doing is we're only looking at 1998 to 2015, before the last El Nino, and we get a real good relationship. Next slide. What happens when we add the last year? 17, 18, 19, 20, 21, 22. We're getting more young green crabs than we were predicting by our original model, right? That green crab larvae are coming from California. Next slide. Southern water mass, strong year class. Okay, again, 98 to 2015, we're looking at how strong the Davidson current is. And when the Davidson current is strong, when we have lots of southern copepods in our water, we get good green crab recruitment. Next slide. And again, we have the outliers after the last El Nino. Another thing we observed was prior to the last El Nino, you know, we'd get a nice bell-shaped curve of sizes of green crabs. You know, they would range from 30 to maybe 55 millimeters in carapace width at the end of their first growing season. In 2018, and in some years, we get lots of little ones here that are less than 30 millimeters in carapace width. And also Christina Geierman, who is a marine biologist and also a very good science teacher at North Bend. When she was walking her dog, she was picking up all these small little green crab moles, and these would have been smaller even after the animal molted. It would be smaller than the crabs. They would be too small to enter our traps. So we're getting a lot more smaller crabs in some years. Next slide. So where are they coming from? This was our old model, the Davidson current bringing larvae, green crab larvae from California. And the longer the Davidson current and the stronger, the more green crabs we got. The currents shift sometimes, you know, in the spring. That's called spring transitions. The winds blow from the north, pushing water southward, bringing all these really yummy copepods to our shores. And that could also be a mechanism for green crab larvae to be transported from the north, from Washington, from British Columbia to Oregon. And since these larvae would settle later in the year because they couldn't arrive before spring transition, so they would arrive after spring transition in the summer, they would be smaller than the ones that came from California. Next slide, please. Okay, so now things are getting a little complicated. At first, we just had one hypothesis, and It was very easy to support it. Larvae arrived from California in the Davidson Current during the winter, and we got more when the winters were warm and the Davidson Current was strong. Hypothesis 2 is that there are now additional larval sources because green crabs have increased in abundance in Oregon, Washington, and British Columbia. So these could also provide larvae for our estuaries. And we do have evidence. Okay, California, we know that. We have local seeding. Alan Shanks at Coos Bay was measuring, he was sampling plankton samples by Jordan Cove in Coos Bay. He had a grant from the Jordan Cove Energy, you know, the LNG plant that they were trying to get started. So he did an environmental impact study looking at what kind of plankton were available year round. And he found that in 2010, he found a very young green crab larvae in January, February, and March of that year. So they could not have come from California. They would have had to be locally produced because they were young, very young. And at that time, they, depending on what the currents were doing, that in the winter they would be taken northward and maybe seeding some of these estuaries, and in the summer the larvae, let's see, if late spring they would be taken southward. Yes. Okay. Also, this is a really interesting story. There's one population on Vancouver Island that's genetically distinct from all the green crabs on the whole West Coast. And you know how that happened? The gene pool is very well mixed, as you can tell, because, whichever way the currents are going, everything gets well mixed. But there were some biologists that were on the West Coast of Vancouver Island. They were collecting mussels for toxicology studies, right? They wanted to see if there was any red tide organisms in the mussels. And they weren't willing to deal with them right away, so they parked them in Souk Basin, Souk Harbor, which is just northwest of Victoria, and they probably forgot about them. And that's how we think green crabs were introduced into Souk Harbor. And these crabs, of course, they had a founder effect, just a few individuals. So they have limited genetic variability, and then they inbreed like hell, and so they have a distinct genetic fingerprint. And what we've been doing is we've been sampling our young of the year crabs. We've been taking little genetic samples. We we sample a foot, like a leg, and put it in a vial with alcohol, and then send it to Carolyn Tephall, who is a geneticist at Woods Hole. And she's been studying green crab genetics since she was a graduate student, so well over 20 years. And so we sent her samples, and she found that from Macaw Bay, Willapa Bay, Tillamook Bay, and Neatcarts Bay, there was at least one animal each that originated from Souk. Can you imagine? It's like finding a needle in a haystack. And that's just an indicator that a whole lot more that are coming from the north, right? I mean, it's just one small population. So that is really interesting. Next slide. Okay, do we have time? I guess we still have some time. What can be done? What can we do about these green crabs? Any ideas?
What are their predators?
What are their predators? Well, birds will eat them. Other crabs, larger crabs will kill them. When they're small, fishes will eat them. Yeah. So they're part of the ecosystem. They have their predators, and however, eradication is no longer on the table. As you can imagine, with something that has larval development, larval gets transported around as larvae. Eradication should have been done in the slough in San Francisco Bay when nobody was looking, right? I mean, nobody was aware. By the time people were aware, in 1990, people noticed that green crabs were in San Francisco Bay, and that's when scientists became involved. But there were already all sizes of green crabs, including gravid females. So it was too late. Once the scientists found out, it was too late to do anything. And what we can do, though, is we can reduce their impact, but we have to define where we want to do our control measures. You know, like eelgrass beds, we should make sure that eelgrass beds don't get torn up. Shellfish beds.
How do you do that? Right. Yeah.
How do you do that?
How do you make sure that they don't tear up the eelgrass bed?
You try to trap them out, but it's limited. You know you can't do, you know, the whole of Yaquina Bay. You can't do it. You know, you just have to decide. Okay, this is a patch I want to control, and you have to keep doing it. And so you know, there's some places where we might want to do some control measures. Next slide, please. And how do we do it? Well, right now we don't have a lot of tools. Trapping seems to be our major tool right now. Another thing is we need to make sure that we keep our ecosystems healthy, and that means that we should not overfish the predators, like, for example, red rock crabs. Red rock crabs might be controlling green crabs in the lower estuary. Fences have been tried. Poison bait's not a good idea. Introducing green crab parasites, they've been looked at that, but these parasites are not species specific, you know, so it's not a good idea. Sex pheromones, I think there could be some promise in the future, and it's been suggested if you can't beat them, eat them, right? How many of you think you can control them by eating them?
You could overfish.
Can you overfish them?
I don't know. Are they tasty?
They're tasty, yes. Apparently, they're fine. They taste fine. People like them. They taste like crab, and you know you can
get a good marketer.
The next best thing.
Next slide, please. Well, green crabs are being controlled in Washington. What happened in Washington was that they started a green crab monitoring network of volunteers called Crab Team. It's sponsored by Washington Sea Grant, the University of Washington. And they got a lot of tribes and shellfish growers and just private individuals involved monitoring green crabs. And when they were found, they would do a rapid assessment. And now the green crabs are at a level where they're being controlled in certain areas. They were controlled at Dungeness Spit, Drayton Harbor, Lummi Sea Pond, and I'm sure they're being controlled by some of the shellfish growers in Samish Bay and Padilla Bay. But anyways, I'm going to talk to you about what's been done at Dungeness Spit and at Lummi Nation Sea Pond. At last year, they found very few crabs at Dungeness Spit, less than 10. Lummi Sea Pond, on the other hand, they found 80,000. Yeah. Okay, next slide, please. Dungeness Spit National Wildlife Refuge. Green crabs got into Dungeness Spit during the last El Nino, the 2015-16 El Nino. And based on the sizes of the crabs that they trapped, that's the year classes that got in through the Strait of Juan de Fuca when the water was warm enough. It was too cold before, and it's too cold after. So there was just a short window of time when the green crabs got through the Strait of Juan de Fuca into the Salish Sea. So now Dungeness Spit is, you know, a rather small area, and they were trapping very intensely. Lawrence Solomon, the manager, bless his heart, he had the idea of controlling them, even though they weren't very abundant. There was 0.5 crabs per trap. Okay, like you put out 100 traps and you get 5 green crabs. So they trap them intensely. They hit them hard, they hit them early. The first few years, they put 5,000 traps per year. And they were trapping every working weekday. And they got it down to 0.01 crabs per trap last year. And they have found no recruits last year. And this year, very similar. They're still finding a few crabs, but not enough that they are going to be a problem, right? They're so rare that you cannot detect any effect. Next slide, please. Now here, this is Bellingham Bay, and this is Lummi Nation land right here. That's a peninsula. And what the Lummi Nation did was they constructed a 3-mile dike to create a pond on their mud flat. They did that in around 1970s. And they were rearing, they're doing aquaculture, salmon aquaculture and shellfish aquaculture in there. But guess what? All it took was a few green crabs getting in there, and they're using that as an incubator. So they removed 80,000 last year. So what they decided to do is, well, actually, what happened was in Washington, a whole bunch of shellfish growers and mainly the Lummi Business Council, they were really influential. So so they had tribes, they had agencies, and they had shellfish growers, and they all approached Governor Inslee and told him that the green crabs are destroying our livelihood, and they need to be controlled. And so he came up with $6 million, $6.5 million for control. And so they're able to hire people to do trapping. And the tribe actually hired a lot of their high school students to do trapping. And what they did was they got, last year they got 4 per trap. Now this year they got 1 per trap. And this year they did not get the recruitment that they saw last year. Very few young of the year were found this year. They're going to keep up the trapping over the winter and then again next year. But they're going to have to keep doing that forever, I would think. Yeah. Next slide. Oh, I mentioned that our native crabs could actually be keeping green crabs in check, but we don't know. We do know that the red rock crab does tend to keep green crabs out of the lower estuary. Here's, like at Port of Toledo, you don't get any green crabs or red rocks because it's too brackish. Riverbend, Rogers Dock, and the Oregon Coast Aquarium, they're sort of mid-estuary, and that's where green crabs thrive, and you don't see any red rocks. But I had a student who actually looked at green crab, red rock crab interactions, and red rock crabs will eat green crabs the same size and smaller versus eating their own kind. They're not as cannibalistic. They will eat green crabs preferentially. So, we should not really, we should keep the red rock crabs, shouldn't reduce their numbers too much. Next one. Also, those little crabs, Hemigrapsis oregonensis and Hemigrapsis nudus, these little shore crabs, they could be doing their job too. You ask, well, how can a small crab can control a big crab? Well, these Hemigrapsis crab have a cousin, Hemigrapsis sanguineus from Asia, that was introduced to the East Coast. And they became quite abundant, especially in structured habitat like riprap. And people were noticing when Hemigrapsis sanguineus was abundant, the green crabs went down in numbers. They were eating the young green crabs, the small green crabs. So don't ever underestimate what these crabs might be doing, because there's a lot of interactions that might be occurring at a very small scale that we don't even think about. Yeah. Next slide, please. Okay, pheromones. I mentioned pheromones. Well, that little crab there is trying to mate with a with a golf ball that has been smeared with female sex pheromone. And you can see that, you know, tactile cues and visual cues don't matter. It's just that scent that drives them crazy. And we set up some experiments with traps like that in Yaquina Bay, right where that natural gas tank is along Sally's Bay. And these are our traps. They have a slit-like opening on the side, and we baited them with real bait, tuna bait, a placebo, a non-scented control, and pheromone, female pheromone, sex pheromone. Next slide. And you can see this is a pheromone one. These are the little tea balls that contain the jelly that contains the pheromone. And then there's the one that has no scent. And you definitely catch more green crabs with pheromone than you do with the no scent control. The pheromones work as well, if not better, than tuna bait. But you need to have something that works even better than that, don't you think? I mean, anyways, let's go on. All right. So in Venice, there is a seasonal fishery for green crabs in the spring and in the fall when they molt. And what these people do, it's a century-old fishery, you know, over 200 years old. And it's like maybe a few families that catch crabs, and then what they do is they examine them. They can tell whether a crab is about to molt or not, because the carapace, the old carapace is being slouched off. It becomes sort of whitish, and those crabs they'll keep and put them in the baskets and watch for them to molt, and then take them to the market. Next slide. And these are soft-shelled crabs in Venice, selling for 48 euros per pound, per kilogram, which is like $22 per pound. It sounds pretty lucrative, but it's a lot of work. unless these crabs all molt at once, you know, you're going to have to, how are you going to do it? You can keep your crabs in a tank and pull out the ones that have molted. Well, they're cannibalistic, you know, and molting is suicidal if you're in a tank with other crabs. So it's not as lucrative as you think. Next slide. This is the green crab cookbook. Next slide. That was put together by a couple of women, Mary Parks and Tan Tai, supported by Monument and New Hampshire Sea Grant. And you know, some of the chefs got really creative. There's some really yummy recipes in there. And I have not, but I've had colleagues that have tried them. They said they taste just fine. They taste like any other crab. One colleague, actually the aquarist at the Hatfield Center, he made crab cakes, but he says he'll never do it again because it's so much work. There was one recipe, it's called greencrab.org. It's one of those recipes. They took it off the webpage. But I remember it called for 20 green crabs to make a broth to make green crab risotto. Okay, 20 to make the broth. And then you needed a pound of crab meat for the topping. Next slide. Can you see yourself doing that?
Wow.
If it takes four Dungeness crab to yield 1 pound of meat. I've done that. But then how many green crabs would it take? So I think you know that there's a use for them in broth. You can make a nice broth with them. And the soft-shelled crab industry, I don't think is there yet. You know, they're trying, but I think it'd be very labor intensive because you'd have to keep the crabs separate. Okay, should we trap and eat green crabs? Yes or no? In California and Oregon, it's yes. You can have 35 crabs per day, but you need a license. In Washington and British Columbia, it's still illegal to possess green crabs. Don't collect them. You can take a photo and send it to the agency that you found it. One reason is that in Washington and British Columbia, there are still places where green crabs have not been found, and they don't want people moving them around. So fishing them will not make a dent into their numbers. I mean, in Newfoundland, they're paying fishermen to fish green crabs, and they're just taking them straight to the dump. They tried making fertilizer out of them, but it costs more for transportation than you get from the fertilizer. It's just not feasible. Okay, you don't want to move them around, such as Hood Canal doesn't have green crabs yet, so you don't want people moving them there. They're not monitored for toxins, for harmful algal blooms, and also green crabs can live in areas that are not safe for shellfish culture. So beware if you want to eat green crabs. Get them from nice clean water. Okay. What do we know? We know we have 8 consecutive strong year classes in the population. Well, would only be 6 because they only live for 6 years, right? But at any rate, we'll have a larval source till 2028, even if we don't get any recruitment from outside. We have multiple larval sources now. Green crabs will become part of the ecosystem. Next slide. But we don't know how abundant they'll become. You know, we saw that initially, you know, between the 2 El Niños, numbers were up and down, really, really low, less than 1 per trap. Then the 2015-16 El Nino, we saw an increase and then maybe a leveling off. Will that continue? We don't know. We've had La Nina years, La Nina conditions for a while, and another La Nina is predicted for the future. However, if we get another strong El Nino, we might just get more than what we have right now. Right now, we have about average of 6 per trap in Yaquina, and in Coos, roughly 4 to 6. At 10, you're definitely going to see some ecological effects. Next slide. And now I can take questions.
So we have a question from one of our online people from Indonesia. Eric is asking, his first question, he has 2 questions. First, is there any case of white spot syndrome virus outbreak for green crab? And what is the chances for using them to, and what is the chances for using them to control, or for using them for biocontrol?
I'm not sure about that white virus.
White spot syndrome virus.
White spot. I don't know anything about that. I'm sorry. And
That was the second. The second question was, and using them for biocontrol. And using it for biocontrol.
well, they've tried biocontrol for green crabs. They introduced, you know, they tested some of the parasites they had in Europe in the lab, and they decided not to do it because the parasite would attack native crabs. Yeah.
It sounded like maybe he was asking about using the green crabs to control something else. You think?
I'm not sure.
I don't understand that question then. Sorry.
If there are some of the things that the green crabs eat that you want to control, introducing them to control something else. Maybe.
I have a question.
Yes.
So with this uptick that we've just seen, especially with northern recruitment coming south, is that potentially because of the blob that we saw, that warmer water that came really far up north?
That blob, in conjunction, the blob was 2014, and then in conjunction with the 2015-16 El Nino, that were like 2 really warm winters. That's when larvae got into the Stray Water Fuca. And yeah, before that, the Strait of Juan de Fuca is pretty cold in the winter. And it's only during the winter storms that that water comes into the Strait of Juan de Fuca. Usually it's estuarine circulation, where, you know, water from the Fraser and the Skagit and other rivers flows out of the Strait of Juan de Fuca. Yeah.
Are those Cannon Beach people likely to see green crabs in the tide pools along the coast here?
Well, so, you know, green crabs have been mainly restricted to the estuaries, the mud flats. But this year, I've had 2 reports of them being on the open coast at Boiler Bay and another spot. So they could be coming into the tide pools. On the East Coast, they are on the open coast. Yes.
So what effect are they gonna have on the Dungeness crabs?
On the Dungeness crabs, young Dungeness and green crabs live in the same habitat. So they're probably eating the same kind of food.
Do you see a decline in the Dungeness?
We haven't seen that yet. You know, our crabs are not that abundant. The green crabs aren't quite that abundant yet that we could detect that. Yeah, we don't know. Yes.
Yeah, so kind of to piggyback off that question, we saw several years ago that ocean acidification and hypoxia around the Oregon coast played a significant role in the decline of the Dungeness crab fishery. And it's kind of through submerged aquatic vegetation, primarily eelgrass beds, which is, you know, habitat for juvenile Dungeness crabs. Is there any research or understanding of how OA age impacts can affect potentially green crabs, or that kind of like top-down predator control from like the red rock crabs or the purple crabs?
I'm sorry, I can't answer that question. I don't know. Yeah, we don't know. I don't know yet. Yeah, but we know that that green crabs, gravid green crabs, when they're in acidic water, they don't clean their eggs. They don't, you know, they don't aerate them and they don't clean them. They're lazy mothers. And so that could be a problem. Yeah. Yeah. Yes.
This is probably a question about crabs in general, but when at the beginning of your discussion, I was thinking, do they get oxygen both from air and from water, whichever they're in?
Right. Especially green crabs can be out of water for quite a while, as long as their gills are wet. You know, there's just one membrane that has to be wet because, you know, the oxygen diffuses across that membrane. Yeah, if they're wet, they'll be fine out of water.
Is that true of other kinds of animals? Obviously, I'm not a very knowledgeable person.
It's true for, I think crabs can live longer out of water if their gills are wet. Yeah, yeah.
And other things besides crabs?
Well,
it's just not a question that ever occurred to me.
A lot of your intertidal animals can, because they have to be able to withstand that tide coming in and out. So not indefinitely, but the same thing, as long as they stay moist. So like barnacles will close their shells so they retain moisture. So a lot of those things can do.
I just assumed that the particles sort of held their breath.
yes.
I'm just curious about, I thought red rock crabs didn't like a sandy environment.
They don't.
But they can live in the same estuary.
They live more where it's more rocky, you know, where the substrate's more rocky. Yes.
Okay.
Yeah, you're absolutely right. You don't. You sometimes you find them, you know, on the mud flat, you know, but I think they move in and out with the tide. I catch them in the same traps as green crabs, and it's usually it's not all sand, not all, you know, all mud.
Okay.
There's some rocks around it. Yeah.
So Tillamook Bay is super muddy and doesn't have a lot of rocks, you're not gonna find the red rock crabs there as much. You'll find a lot more green crabs there, but if you go into parts of Netarts where the rocky jetties are, it's full of red rock crabs. Same with the seaside cove, just full of red rock crabs. So it kind of just depends on that habitat. Make sure they're.
We're a great audience. So many good questions. And it's nice to meet in person. It's a real treat, you know? I'm so tired of Zoom.
We are too.
Well, thanks everybody for coming. Feel free to grab a